Let me ask you a question. Right now, what is your blood type?
I mean, there is a very good chance that when you hear that question, you just think of a simple letter and a mathematical symbol,
Right.
right?
Yeah, exactly. Maybe you are A positive, or, maybe you are O negative or AB positive. And for most of us, honestly, that's where the understanding stops.
Right. It's just a category in a medical chart.
Exactly. It's a detail you might need if you ever end up in an emergency room or a box you check when you go to donate blood. But what if you were told that your blood type is actually this insanely complex microscopic molecular barcode?
An evolutionary fingerprint, really,
Ah,
one that tells a story spanning thousands of years.
right. What if I told you that these simple letters represent highly specific architectural structures? Structures that basically dictate everything from your statistical risk for heart disease to how your body reacts to a microscopic, mind-altering parasite.
It's wild. It fundamentally rewrites our relationship with our own biology. I mean,
Yeah.
we grow up picturing blood as this homogeneous red fluid, essentially serving as just a simple liquid transport system.
Like plumbing.
Yeah, exactly like plumbing. But when we zoom down to the cellular level, we are looking at a highly customized, genetically inherited ecosystem. It's this bustling, microscopic environment that's been shaped by, thousands of generations of environmental pressures.
And that is exactly what we are setting out to do today. Welcome to the deep dive. We have a towering stack of material in front of us today, spanning genomics, advanced biochemistry, evolutionary biology, and metabolomics.
It's a lot of ground to cover.
It is, but our mission is to synthesize all of it. We are going to map out the molecular linkages, the genetic quirks, and the biological tangents that make up this system. Because usually, knowing your blood type is sort of like knowing the make and model of a car.
Right, you drive a Honda Civic.
Yeah, but today, we are tearing down the engine block to the molecular level. We're going to completely redefine what blood actually means to you.
To properly frame this, I think it helps to understand how relatively new this whole concept is. I mean, before the turn of the 20th century, the prevailing medical consensus was essentially that all blood was identical.
A red fluid was a red fluid.
Exactly. And this was a profoundly dangerous misunderstanding. Doctors were actively attempting animal-to-human transfusions like using sheep or dogs.
Which sounds completely insane to us now.
It was a nightmare. Or they tried these hazardous transfusions between people.
Yeah.
And the results were frequently catastrophic. The patient's blood would just clot inside their veins, their urine would turn pitch black, their kidneys would fail.
And no one understood why.
Right, no one had a clue. It wasn't until Carl Landsteiner's pioneering work in the early 1900s that the mystery began to unravel. He essentially proved that human blood isn't a universal substance at all.
So he transitioned our understanding from this mysterious bodily fluid to a genetically inherited, highly specific system of antigens and antibodies.
Yes.
Okay, let's unpack that transition. Because to really understand how blood types determine our health and why someone else's blood might be biologically toxic to you, we really have to understand what these blood types physically look like.
We have to move from the macroscopic liquid in your veins to the surface topography of a single red blood cell.
Right. And the foundational biological concept we have to tackle here is the antigen.
Right, so in classical immunology, an antigen is basically defined as any substance that your immune system recognizes as foreign.
Something that prompts it to produce antibodies.
Exactly. Now, on the membrane of a red blood cell, antigens act as distinct surface markers. But the critical nuance, and this is what often gets lost in high school biology, is that these markers aren't all made of the same raw materials.
The biochemistry building these structures is actually incredibly diverse and, well, really elegant.
It is.
Let's start with the most famous one, right? The ABO system. So A, B, AB, and O. Because what immediately stood out in the source material is that ABO antigens are not proteins.
No, they aren't.
When we think of genetics, we almost always think of DNA coding for proteins. But these are sugars, specifically oligosaccharides.
That is a vital biochemical distinction to make right out of the gate. DNA does not directly code for sugars, period. The central dogma of molecular biology is that DNA transcribes to RNA, which translates into proteins.
Right, the standard pipeline.
So the blood group antigens in the ABO system are what geneticists actually call secondary gene products. The primary gene products, the actual proteins encoded by your DNA, are a class of enzymes called glycosyltransferases.
Okay, I want to make sure we're visualizing this correctly. Let me try an analogy here. It's like your genes don't actually build the house, meaning the final antigen itself. Instead, your genes build a set of highly specialized robotic arms. Those robotic arms are the enzymes. And it's those robotic arms that are programmed to grab specific bricks, the sugars, and stack them on top of each other in a precise sequence.
That's a very useful way to picture it, yeah. The genetic code provides the exact blueprint for the robotic arm. Depending on which blueprint you inherited from your parents, your robotic arms will grab very specific sugar molecules and link them together in highly specific geometrical arrangements.
But to understand this construction process, we have to look at the foundation they're building upon, right?
Exactly. Every single A, B, and O antigen starts with a base structure called a precursor oligosaccharide chain. And there are two main types of these precursor chains, simply named type 1 and type 2.
And the difference between type 1 and type 2 just comes down to a tiny, almost imperceptible chemical bond, right?
It does. It comes down to stereochemistry,
Yeah.
so the three-dimensional arrangement of the atoms. Both of these precursor chains end with two specific sugars: a galactose molecule attached to an N-acetylglucosamine molecule. In a type 1 chain, the linkage between these two is what biochemists call a gal beta 113 GLC-ANC bond. That means the first carbon atom of the galactose is chemically linked to the third carbon atom of the N-acetylglucosamine.
Okay, so carbon 1 connects to carbon 3.
Correct. But in a type 2 chain, it's a gal beta 144 GLC-ANC linkage. The connection is on the fourth carbon atom instead of the third.
I mean, that sounds like a trivial geometric shift, like plugging a USB cable into a slot that's just a millimeter to the right. Does it really matter?
Oh, in biology, structure dictates function entirely. That tiny geometric shift changes the entire physical shape and angle of the molecule jutting out of the cell. If you plug that USB in at a different angle, the casing just won't fit.
That makes sense.
And for our purposes today, type 2 chains are the specific physical structures primarily found jutting out of the lipid bilayer membrane of your red blood cells.
So we have this type 2 precursor chain sticking out of the red blood cell like a bare antenna. But that's not a blood type yet. It's just the raw material. This is where the first robotic arm comes into play to build what's called the H antigen foundation. Walk me through that.
So to build an ABO blood type, your body first has to construct this H antigen. The blueprint for this is controlled by a gene called FUT1, which is highly active in your hematopoietic tissues, so the bone marrow tissues manufacturing your red blood cells.
Okay, FUT1.
The FUT1 gene produces our first robotic arm, an enzyme called alpha-1-cell-2 fucosyltransferase. This enzyme's sole biological purpose is to grab a specific sugar molecule called fucose and physically attach it to the terminal galactose of that bare type 2 precursor chain.
And once it does that,
The moment that fucose sugar is attached, the entire structure is officially designated as the H antigen.
so the H antigen is the finished basement of the house. You absolutely have to have it before you can build the first or second floor.
Precisely. It is the mandatory docking station. Now, if you happen to have blood type O, the biochemical construction process stops right there. Your red blood cells are released into your bloodstream, completely covered in millions of these unmodified H antigens.
But let's say you have the A allele in your DNA.
Right, then your body goes a step further. It produces a second, entirely different robotic arm. This one is an enzyme called N-acetylgalactosaminyltransferase.
I'm glad you had to pronounce that and not me.
It is a mouthful, I know. But biochemical nomenclature is actually beautifully descriptive if you break it down. It transfers an N-acetylgalactosamine sugar molecule, and it attaches that sugar to the H antigen docking station using an alpha-1 servinode linkage.
And that specific addition creates what we call the A antigen?
Exactly. Alternatively, if you have the B allele, your DNA produces a slightly different enzyme, a galactosyltransferase. This robotic arm grabs a regular galactose sugar and attaches it in that exact same alpha-1 delta position on the H antigen. That creates the B antigen.
Okay, I'm tracking this. If you are type A, your robotic arms stacked the A sugar on top of the H foundation. If you are type B, your robotic arms stacked the B sugar on top of the H foundation. And if you are AB, you inherited the genetic blueprints for both robotic arms, so you have a mix of both A and B sugars heavily decorating your cells.
That's it, perfectly.
But wait, I have to push back on the type O scenario. We just said that if you are type O, the process stops at the H antigen. Why? Is the type O gene actively telling the cell to stop building?
No, it's actually the exact opposite. It's not an active instruction, it's a silent failure. Type O is essentially a genetic deletion mutation.
A mutation?
Yeah. The specific gene that is supposed to manufacture that final robotic arm, the one that adds the A or B sugar, is missing a crucial piece of its sequence. Because of that missing piece, the enzyme it produces is structurally misfolded and completely inactive.
So it's a dead robot. It just floats around, unable to grab any sugars. But doesn't that mean type O blood is technically a broken genetic trait? It's literally a loss of function at the cellular level?
In strict biochemical terms, yes, it is an absolute loss-of-function mutation.
Yeah.
The transcription happens, but the resulting machinery is useless. The H antigen is left bare.
But here is where we have to separate cellular mechanics from evolutionary success. Calling it broken implies a disadvantage. Yet, type O is the single most common blood type in the human population globally. Evolution overwhelmingly favored this broken trait.
Which implies that not having those extra A or B sugars on your cells actually conferred some massive survival advantage to our ancestors, that the dead robot was actually saving their lives somehow.
Exactly. We will explore the specific environmental pressures that cause that later, but it's a brilliant example of how a molecular loss can be an evolutionary gain.
That's fascinating. Okay, so that covers the ABO system, these intricate towers of sugars meticulously built by enzymes. But when anyone asks for your blood type, there's always a positive or negative sign attached to it, right? A positive, O negative.
Yes, the Rh factor.
That brings us to the Rh system. And structurally, this is a completely different beast than the sugar forest we just talked about. It is an entirely different class of molecule. If the ABO system is a dense forest of delicate sugar trees on the cell surface, the Rh system is a network of massive structural protein pillars embedded directly into the cell membrane.
So it's not a sugar at all.
No. The Rh antigens are polypeptides, long chains of amino acids. They are not secondary products built by enzymatic robotic arms. They are primary products directly translated from your DNA transcripts, specifically the RHD and RHCE genes.
And the architecture here is intense from what the sources describe. It's not just a little marker floating like a buoy on the surface of the cell. It's a massive structural complex.
The physical reality of the Rh complex is staggering, honestly. It is a core tetramer, meaning it's composed of 4 massive subunits. You have 2 Rh protein subunits and 2 closely associated glycoprotein subunits called RHG.
Okay.
And these proteins don't just sit on top of the lipid membrane. They are multi-pass transmembrane proteins. They weave in and out of the cell membrane 12 separate times.
12 times, like sewing a thick thread back and forth through a piece of fabric.
Yes, exactly, to ensure it is incredibly secure. Furthermore, this entire tetramer complex doesn't just float freely in the membrane. It is non-covalently bound to the red blood cell's internal skeleton.
The internal skeleton.
Yeah, inside the red blood cell is a complex lattice of structural proteins called the actin-spectrin cytoskeleton. The Rh complex is physically tethered to this internal scaffolding via anchor proteins called protein 4.2 and anchorin, which connect directly to band 3, a major anion exchanger in the cell.
Let me make sure I understand the mechanics of this. It's essentially bolted to the internal scaffolding of the red blood cell. So it's actually providing structural integrity to the cell itself.
That is its primary physical role. Red blood cells have a brutal existence. They are constantly being pumped through massive arteries at high pressure and then squeezed through microscopic capillaries that are actually narrower than the diameter of the cell itself.
Right, they have to squish and fold.
They have to deform, stretch, and bounce back constantly. If they didn't have massive structural pillars like the Rh complex bolted to their internal skeleton, the sheer stress of blood flow would just rip them apart.
Okay, so if you are Rh positive, you have the RHD gene, and you build this massive D antigen structural pillar. If you are Rh negative, what happens? Are you just walking around with fragile, easily torn red blood cells?
Not quite. If you are Rh negative, you simply lack the RHD gene. You don't build the specific D antigen protein. However, you still possess the RHCE gene and the RHG glycoproteins.
Oh, so the structure is mostly still there.
Right. The structural complex is still formed and anchored. It's just missing one specific molecular component, the D antigen loop that protrudes outward. So the structural integrity of the cell is largely maintained. We'll discuss what happens when the entire complex goes missing a bit later, and it's quite catastrophic.
I want you, the listener, to take a second and visualize the surface of your own red blood cells right now. They aren't these smooth, perfectly round inner tubes like the simple textbook drawings suggest.
Not at all.
They are dense, chaotic, microscopic metropolises. You have these sprawling, branching canopies of ABO sugars reaching outward, and right next to them, you have these massive, multi-pass protein pillars of the Rh complex bolted directly into the cell's internal skeleton.
And every single structure in that metropolis, every single sugar bond and protein weave was dictated by the specific genetic blueprints you inherited, which naturally leads us into the mechanics of that inheritance. How do these blueprints actually combine to build you?
Right. Let's look at Mendelian inheritance. This is usually taught with those classic high school biology Punnett squares, but let's take it out of the textbook and see how the mathematical reality of it works. The ABO gene is located on chromosome 9.
Yes. Like almost all chromosomes, you inherit one copy of chromosome 9 from your mother and one copy from your father. Because there are three main alleles in the human gene pool for this system, A, B, and O, there are exactly 6 genetic combinations or genotypes you could possibly possess.
Right. AA, AO, BB, BO, AB, or O.
Exactly. But the phenotype, what actually manifests in your blood, only have 4 categories, because A and B are completely dominant over O.
So if I have the AO genotype, my blood just tests as type A. The O is basically silent.
Correct.
But what happens with the AB genotype? Why doesn't A overpower B or vice versa?
That introduces the concept of codominance, which is a beautiful genetic compromise. If a child inherits an A allele from one parent and a B allele from the other, neither gene masks the other.
They both just go to work.
Exactly. The cell's machinery reads both blueprints and diligently manufactures both types of robotic arms. The N-acetyl-galactosaminotransferase for A and the galactosubtransferase for B. Both enzymes operate simultaneously in the Golgi apparatus of the cell, frantically attaching their respective sugars to the available H antigens.
The result is a red blood cell completely covered in a mix of both sugar chains. If we look at the mathematical matrix of inheritance, mapping the 4 possible maternal blood phenotypes against the 4 paternal ones, we get 16 possible combinations. And the core rule that jumps out from the sources is the inheritance of type O.
Yes, the math is very rigid there.
An O parent can only pass an O allele to their child. Because to express type O blood, you mathematically must be O genetically. You have no A or B blueprints to give.
That mathematical certainty has historically been used in everything from forensic science to paternity testing. But there is a massive conceptual leap we need to make here. The genetic inheritance of these sugar-building enzymes isn't just limited to constructing red blood cells.
Right.
The blueprint isn't exclusive to the bone marrow. This bridges hematology with gastroenterology and immunology. We really need to explore the secretor system.
This was the part of our research that fundamentally shifted my perspective. The idea that my blood type isn't just trapped inside my veins, it's in my spit, my sweat, my tears, and my stomach acid. Walk us through how that is physiologically possible.
It comes down to a parallel genetic pathway. Earlier, we talked about the FUT1 gene. That is the gene that builds the H antigen foundation on red blood cells using type 2 precursor chains.
Right.
Well, there's a second closely related gene called FUT2, historically known as the SAZ gene. FUT2 produces an enzyme that is essentially a biochemical twin to the FUT1 enzyme. But here is the critical difference. FUT2 does not operate in the bone marrow. It operates exclusively in your secretory glands.
By secretory glands, we mean salivary glands, the mucosal lining of the gastrointestinal tract, the respiratory tract.
Yes, your tear ducts, your mammary glands producing breast milk, and even seminal fluid. If you inherit the dominant, say, allele of the FUT2 gene, you are classified as a secretor.
And what does that mean practically?
This means your secretory glands are actively manufacturing H antigens, and depending on your ABO type, A and B antigens, they are constantly shedding these highly specific blood-type sugars directly into your bodily fluids.
What percentage of the population does this?
Roughly 80% of the human population are secretors. If you are a type A secretor, your saliva is literally packed with type A sugar antigens. A forensic scientist could determine your blood type just from the saliva left on a coffee cup.
That's wild.
But there is a subtle biochemical nuance. The FUT2 enzyme in your glands primarily acts upon type 1 precursor chains, whereas the FUT1 enzyme in your bone marrow acts on type 2 precursor chains. Remember that tiny shift in the carbon linkage we discussed earlier?
The gal beta-1-ethyl-3 versus 1,4,4 bond, the USB cable plugged in at a different angle.
Exactly. The sugars in your saliva have a slightly different three-dimensional geometry than the sugars on your red blood cells, even though they represent the same blood type.
And what about the other 20% of people, the ones with the recessive C's genotype?
They're termed non-secretors. Their bodies still build the antigens perfectly fine on their red blood cells via the FUT1 gene in the marrow. But because their FUT2 gene is inactive, their bodily fluids and mucosal linings are completely devoid of ABO blood group antigens.
Why does this matter? If I'm a non-secretor, does it affect my health?
Profoundly. We will delve into the microbiome later, but as a quick teaser, those sugars in your mucus are primary binding sites for bacteria and viruses.
Like chemical handles.
Exactly. For instance, norovirus, the notorious winter vomiting bug, specifically binds to the H antigen in the gut mucosa. Many non-secretors are virtually immune to common strains of norovirus because their gut lining lacks the specific sugar handle the virus needs to grab onto.
That is incredible. But wait, I want to clarify something about testing this. The material mentions the Lewis blood group system in relation to the secretor status. How does the Lewis system act as a tell for whether you are a secretor or not?
This is an elegant biochemical logic gate. The Lewis system is controlled by a third gene, FUT3. FUT3 produces an enzyme that adds a fucose sugar in an alpha-1,4 linkage to the subterminal sugar of a type 1 chain.
Okay, stick with me here. Let's trace the logic.
If you are a non-secretor, your type 1 chains in your glands are bare. FUT3 comes along, adds its fucose, and creates what we call the Lea antigen.
Okay, so non-secretors produce the Lea antigen in their saliva.
Right. But if you are a secretor, your FUT2 enzyme has already been at work. It has already added a fucose in the alpha-1,2 position.
Right.
So when FUT3 arrives and adds another fucose to that exact same chain, the resulting double fucosylated structure changes shape. It is no longer recognized as Lea. It is now classified as the Leb antigen.
So simply by taking a cheek swab and testing someone's saliva for the presence of Lea or Leb antigens, a lab can entirely reverse engineer their secretor genetics. If they have Leb, they must be a secretor. If they only have Lea, they are a non-secretor.
It is a perfect biochemical cascade.
Speaking of genetics being locked in, I want to explore a mind-bending caveat. We are taught that your blood type is the ultimate immutable biological identifier. It is inherited for life. Except there are rare instances where your blood type can fundamentally change.
Yes. Blood type chimerism. There are temporary instances caused by severe infections or malignancies. Certain types of leukemia can suppress the expression of ABO antigens, making a type A person temporarily appear as type O. And some bacterial infections can enzymatically alter the A antigen to look like the B antigen, a phenomenon known as acquired B. But the most profound, permanent alteration to a human's blood type occurs following an allogeneic bone marrow transplant.
Explain how that physical transformation takes place.
Yeah.
If I am type O and I receive bone marrow from a type A donor, how does my entire circulatory system rewrite itself?
It is basically a matter of replacing the factory. Hematopoietic stem cells, the master cells responsible for constantly manufacturing your blood, reside exclusively in the bone marrow.
Right.
During a transplant procedure for something like leukemia, the patient's original disease bone marrow is intentionally eradicated. This is done through massive doses of radiation or chemotherapy. The patient's native blood manufacturing factory is completely demolished.
But they still have their own red blood cells circulating in their veins, right?
They do, for a short while. Red blood cells do not have a nucleus. They cannot divide or repair themselves. They have a strict biological lifespan of about 120 days. Once the patient's marrow is destroyed, the donor's healthy stem cells are intravenously introduced. These new stem cells migrate to the hollow spaces in the patient's bones, engraft, and begin setting up a new factory.
And this new factory is reading the genetic blueprints of the donor.
Exactly. The new stem cells carry the genetic code of the type A donor, so they begin aggressively manufacturing type A red blood cells. Over the next few months, the patient's original type O red blood cells reach the end of their 120-day lifespan, enter the spleen, and are dismantled.
And they are replaced entirely by the new type A cells.
Yes. The patient's native immune system also reboots to tolerate the new blood. Genetically and phenotypically, the patient truly and permanently becomes type A.
That is basically medical alchemy. Okay, we've established the complex rules. We understand how the molecular robots stack the sugars, how the structural protein pillars are bolted into the cellular cytoskeleton, and how these blueprints are inherited and sometimes secreted.
We have the foundation.
Now we have to look at what happens when these biological rules are broken. Because this is where blood typing stops being a fascinating biochemical parlor trick and becomes a matter of sudden, violent death.
Yes. To understand the violence of a mismatched transfusion, we have to return to Carl Landsteiner. He discovered what is now known as Landsteiner's law.
And what is that?
The human immune system operates on a very strict binary rule regarding the ABO blood group. If an individual naturally lacks an A or B antigen on their cells, their immune system will continuously produce the corresponding antibody in their blood plasma.
Let me make sure I have the logic. If I am type A, my cells lack the B antigen. Therefore, my immune system acts like a hypervigilant security apparatus, constantly churning out anti-B antibodies, patrolling my bloodstream, just waiting for the B antigen to show up so it can attack.
Yes, it does this naturally, without ever having been exposed to type B blood previously. This is unique to the ABO system.
How is that possible if it's never seen it?
We believe these naturally occurring antibodies develop in early infancy because the bacteria in our gut have sugars on their surfaces that perfectly mimic the A and B antigens. Our immune system sees these bacterial sugars and builds a standing army against the ones we don't possess.
So if a doctor in a chaotic emergency room accidentally transfuses a bag of type B blood into my type A body, what actually happens at the microscopic level? Why is it so catastrophic?
The consequences are almost instantaneous and completely devastating. We need to look at the physical architecture of these antibodies. The antibodies produced against the ABO system are primarily a class of immunoglobulins called IgM.
Describe what an IgM molecule looks like.
It is massive. If a standard antibody is a single Y shape, an IgM molecule is a pentamer. It looks like a massive five-pointed snowflake. And at the tips of all 5 points are highly specific binding sites that act like biological superglue, specifically tuned to the B antigen.
So when this massive snowflake encounters the foreign red blood cells from the transfusion, what does it do?
Because of its enormous size and multiple arms, one single IgM molecule can grab onto multiple foreign red blood cells simultaneously. It cross-links them. Millions of these IgM snowflakes are doing this at once, rapidly gluing the foreign red blood cells together.
Wow.
This causes visible agglutination. Inside your veins, the red blood cells literally clump together into massive, sticky masses.
Which would immediately clog up your capillaries and stop blood flow, but I assume that's only half the problem.
It's actually the less lethal half. The mechanical blockage is bad, but the biochemical cascade is what kills the patient. When these massive IgM antibodies bind to the antigens on the surface of the cell, their structure shifts.
Like a trigger.
Yes, this conformational change acts as a flare gun, triggering one of the most violent destructive pathways in human biology, the complement cascade.
I read about this. The complement cascade sounds like a biochemical self-destruct sequence. How does it work?
The complement system is a series of specialized proteins circulating in your plasma, usually designated C1 through C9. When the IgM antibody binds to the cell, the first protein, C1, docks onto the stem of the antibody.
Okay.
This activates C1, causing it to slice apart C4 and C2. The fragments of C4 and C2 combine on the surface of the cell to form an enzyme called C3 convertase.
It's a chain reaction, an assembly line of destruction.
Exactly. C3 convertase slices up hundreds of C3 proteins, coating the foreign cell. This eventually triggers the final phase, where proteins C5b, C6, C7, C8, and multiple C9 proteins assemble together into a physical cylinder. This cylinder plunges directly into the lipid membrane of the red blood cell. It is called the membrane attack complex, or MAC.
It physically drills a hole through the cell's armor.
It punches a massive pore right through the membrane. Once that hole is created, the cell cannot maintain osmotic pressure. Water and sodium rush violently into the red blood cell. The cell swells immensely and explodes. This is called intravascular hemolysis.
And when millions of cells are exploding simultaneously in your bloodstream,
The sudden, massive release of all that internal cellular debris, toxic free iron and raw hemoglobin into the plasma, completely overwhelms the system. The kidneys attempt to filter out the massive hemoglobin load, but the proteins precipitate in the renal tubules, causing acute kidney failure.
so your kidneys shut down.
Yes, and the shock drops blood pressure to lethal levels. Disseminated intravascular coagulation occurs, meaning the blood clotting system goes entirely haywire, causing massive bleeding elsewhere. Without immediate intervention, the patient will die.
This exquisitely violent reaction is why blood compatibility is so vital. But as I was reading the sources, I came across a medical anomaly that throws a massive wrench into standard compatibility testing. It essentially acts as a biological booby trap for doctors.
Let me guess.
It was first documented in 1952 in India, the Bombay phenotype.
Ah, yes. The Bombay phenotype, genetically designated as H. It is one of the most fascinating and dangerous anomalies in all of hematology. To truly grasp why it's so dangerous, we have to refer back to our earlier house building analogy. Remember the FUT1 gene?
Right. The gene that builds the alpha-1-2 fucosyltransferase enzyme, the crucial first robotic arm that attaches fucose to the bare chain, creating the H antigen. We called it the finished basement that you absolutely must have before you can build the A or B sugars on top.
Keep that in mind. In individuals with the Bombay phenotype, they've inherited two completely recessive H alleles. They possess a specific crippling point mutation in the FUT1 gene. The most classical and widely studied example of this is called the TYR36 terror mutation.
Let's break that scientific jargon down. What does TYR36 terror actually mean at the DNA level?
Well, TYR stands for tyrosine, which is a specific amino acid. 3D16 refers to its exact sequential position in the long protein chain that makes up the enzyme. TER stands for termination, which in genetics means a stop codon.
So normally the DNA sequence tells the cell to place a tyrosine at position 316 and keep building, but a mutation has essentially placed a massive stop sign right in the middle of the blueprint.
That is precisely what happens. During translation, the ribosome is moving along the mRNA tape, assembling the enzyme amino acid by amino acids. It reaches position 316, hits that mutated stop codon, and the entire assembly process prematurely halts.
The robot is half built.
Exactly. The resulting enzyme is truncated. It is missing its final 50 amino acids. Because of this massive structural defect, the enzyme is completely dead. It has zero catalytic activity.
So the body attempts to build the robotic arm, but it comes off the assembly line missing its hand. It cannot grab the fucose sugar.
And because it cannot attach that fucose, the H-antigen foundation is never constructed. The precursor chains on the surface of the red blood cell remain entirely bare.
But wait, here's where I need to logic this out. If the H-antigen foundation is never built, what happens if this Bombay individual actually inherited the strong dominant gene for type A or type B from their parents? Do they just not have those blueprints?
No, they absolutely do. A Bombay individual might have perfectly functioning A or B robotic arms. Their cells might be churning out immense amounts of N-acetylgalactoseminyltransferase.
The A enzyme.
Right, but those A and B enzymes absolutely require the H antigen to act as a physical docking station. They cannot attach their specific sugars to a bare precursor chain. It doesn't fit in their chemical grasp.
So they have the robotic arms to build the roof, but because the basement was never poured, the arms just float around uselessly inside the cell.
Exactly. The final result is that on the surface of their red blood cells, there are absolutely zero H antigens, zero A antigens, and zero B antigens. The cell is functionally naked.
And this leads us to what we can call the transfusion trap. Imagine a Bombay individual gets into a severe car accident and is rushed into a trauma bay. The doctors draw blood and run a standard, rapid serological test. What does that test show?
A standard rapid ABO blood test is primarily looking for the presence of A and B antigens. The lab tech mixes the patient's blood with anti-A and anti-B reagents. Since the Bombay patient's cells have neither A nor B, there is no agglutination. The test comes back negative for both.
And so they assume.
The lab technician confidently signs off on a chart, blood type O, because normal type O blood also lacks A and B.
And that is the lethal trap. They look exactly like universal type O on a standard rush test, but they are not type O. And we have to go back to Landsteiner's law. You produce massive amounts of antibodies against any antigen you lack.
Correct. A normal type O person lacks A and B, so they naturally manufacture anti-A and anti-B antibodies. But their cells are completely covered in H antigens.
Right.
The Bombay individual, however, lacks A, B, and H. Therefore, their hypervigilant immune system produces anti-A, anti-B, and a staggering, highly potent amount of anti-H immunoglobulins.
So if the trauma doctor reads type O on the chart and rushes in a bag of universal donor type O blood,
it would trigger an immediate, massive hemolytic transfusion reaction because that bag of normal type O blood is absolutely coated in millions of H antigens. Yeah. The Bombay patient's powerful anti-H antibodies would immediately recognize the H antigens as a foreign threat, bind to them, trigger the complement cascade, and destroy the transfused blood, likely killing the patient. A Bombay individual can only safely receive blood from another Bombay individual.
the logistics of that are terrifying. The data indicates this phenotype occurs in about 1 in 10,000 people in specific regions of India, but globally, in places like Europe, the prevalence drops to roughly 1 in 1 million.
It's incredibly rare.
If you have the Bombay blood type, you essentially have to freeze and bank your own blood well in advance because finding a compatible random donor in a sudden emergency is functionally impossible.
It perfectly illustrates why modern hematology must prioritize advanced molecular genotyping over simple surface serological testing. We can't just rely on what the outside of the cell looks like. We have to know the actual genetic blueprint operating inside it to ensure safety.
Let's pivot slightly and look at the RH system, because there is an equivalent, perhaps even rarer, nightmare scenario there. We talked earlier about how the RH complex is a massive structural pillar. a multi-pass tetramer, bolted to the internal cytoskeleton to hold the cell membrane together.
Right.
Most people are Rh positive or Rh negative, meaning they either possess the D antigen protein or they don't. But what if the entire pillar is missing?
You are referring to the rhinal phenotype. It is frequently referred to in the media and medical literature as golden blood, because it is arguably the rarest blood type currently known on Earth.
How rare is it?
Fewer than 50 people have been identified with it globally in the last half-century.
Wow.
Yeah. In a standard Rh-negative person, as we discussed, they are just missing the Rhd protein. The Rhce proteins and the Rhe glycoproteins are still present. The structural pillar is still largely functional.
Right.
But in rhinal individuals, due to severe homozygous genetic mutations, often in the RHA gene, they lack all Rh antigens entirely. Rhd, Rhce, everything. The entire complex fails to assemble in the membrane.
If that complex is essentially the bolts holding the membrane to the internal skeleton of the cell and those bolts vanish, what actually happens to the physical structure of the cell? How does it survive the turbulent water pressure of the human bloodstream?
It struggles immensely. Without that anchoring pillar, the structural integrity of the lipid bilayer collapses. The red blood cells cannot maintain their standard, flexible, biconcave disc shape. They deform.
They lose their shape.
Yes, they take on abnormal, spherical shapes or develop jagged, spiked edges, a clinical condition called stomatocytosis.
So they look like spiky microscopic sea urchins instead of smooth inner tubes.
Yes, and because the membrane is structurally compromised, it suffers from severe osmotic fragility. The cell membrane can no longer properly regulate the flow of water and ions across its barrier.
So they just pop.
Pretty much. The cells swell and burst incredibly easily under the mechanical stress of navigating the capillaries. This leads to a state of chronic hemolytic anemia of non-immune origin. Their red blood cells simply have a drastically shortened biological lifespan because they lack the physical scaffolding to survive the journey.
It's incredible that a human body can adapt and function with cells that fragile. And just like the Bombay phenotype, a normal person's immune system will violently attack any Rh antigen.
Absolutely.
So they can only receive blood from another rhinal person, making transfusions a logistical impossibility in an emergency. But the dangers of the Rh system aren't just limited to transfusions between adults. It is historically infamous for complicating pregnancy. This is where hemolytic disease of the newborn, or HDN, comes into the picture.
Yes. The Rh factor was actually discovered in 1939 by scientists Philip Levine and Rufus Stetson, specifically because of a devastating case of HDN. They were investigating a medical mystery where a woman gave birth to a stillborn child and subsequently suffered a severe near-fatal transfusion reaction when doctors gave her a blood transfusion directly from her husband.
How does a mother's body suddenly become toxic to her husband's blood just by being pregnant? I always thought the placenta was an impenetrable barrier between the mother and the fetus.
Well, the placenta is an excellent barrier, but it's not perfectly impenetrable, especially late in pregnancy and during the physical trauma of childbirth. The core issue here is maternal-fetal genetic incompatibility.
Okay, lay it out for us.
If an Rh-negative woman who lacks the massive Rhd pillar becomes pregnant with an Rh-positive fetus, meaning the baby inherited the dominant Rhd gene from the father, a biological conflict is set in motion. During childbirth, the placenta often tears slightly, allowing a small volume of the baby's Rh-positive red blood cells to hemorrhage into the mother's maternal bloodstream.
And the mother's hypervigilant immune system suddenly encounters this massive Rhg protein pillar that it has never seen before.
Right. It recognizes it as a foreign invader and starts building biological weapons against it.
That's terrifying.
This process is called maternal sensitization. The mother's immune system studies the foreign D antigen and produces specific anti-D antibodies. Now, for that first pregnancy, the baby is usually perfectly fine because the sensitization process takes weeks to fully ramp up. By the time the mother has an army of antibodies, the baby has already been safely born.
Oh, thank goodness.
But the danger lies in the immunological memory. The mother's immune system creates a permanent reserve of memory B cells that produce a specific class of antibodies called IgG.
Ah, IgG. We talked extensively about IgM earlier, the massive 5-point snowflake that causes immediate massive clumping in adult transfusions. What makes IgG different in this context?
The crucial difference is physical size and evolutionary purpose. IgM is a massive pentamer. It physically cannot cross the placental barrier. It's simply too big to fit through the cellular tight junctions.
Okay.
But IgG antibodies are single, highly compact, Y-shaped molecules. Evolution specifically designed IgG to easily cross the placenta so a mother can transfer passive immunity to her fetus.
But in this specific scenario, that evolutionary design is turned into a weapon if the mother gets pregnant with a second Rh-positive baby.
Her immune system immediately recognizes the microscopic traces of the baby's Rhd antigen. It aggressively ramps up production, flooding the maternal circulation with these tiny, potent anti-IgG antibodies. They effortlessly cross the placenta, enter the fetal bloodstream, and relentlessly bind to the baby's red blood cells.
And then what? Does it trigger the complement cascade and blow the cells up like a transfusion reaction?
Actually, it's a different mechanism of destruction. IgG doesn't efficiently trigger the massive complement cascade in this scenario. Instead, the IgG antibodies coat the baby's red blood cells, acting like chemical tags that say, "Destroy me." As these tag cells circulate through the baby's own spleen, the spleen's specialized macrophage cells recognize the tags and aggressively devour the red blood cells. The baby's own immune organs destroy its blood supply.
That is brutal.
It causes severe, profound fetal anemia. To compensate, the baby's liver and spleen swell massively as they try to churn out new blood. If left untreated, it leads to heart failure, severe brain damage from bilirubin buildup, or fetal death.
That is absolutely terrifying. It's a biological tragedy. But this is one of those areas where modern medicine has achieved an almost miraculous victory, right? We essentially solved this with prophylaxis.
Yes, the development of Rh immune globulin in the late 1960s is arguably one of the greatest preventative triumphs in modern immunology. Today, if we know an expectant mother is Rh negative, we routinely give her an intramuscular injection of Rh immune globulin at about 28 weeks of pregnancy and again immediately after childbirth.
Wait. Rh immune globulin, which is essentially a concentrated dose of synthetic anti-IgG antibodies, right? If the problem is anti-D antibodies attacking the baby, why on earth are we injecting the mother with anti-D antibodies? Doesn't that just do the damage for her?
It seems highly counterintuitive, I know, but it's a brilliant biological trick. We give her a very small, highly calculated dose. If any of the baby's Rh-positive cells leak into the mother's bloodstream, these injected synthetic antibodies immediately hunt them down, bind to them, and effectively mask or clear them from circulation before the mother's own immune system ever gets a chance to notice them.
Oh. We are essentially cleaning up the crime scene before the maternal immune system's detectives arrive.
Precisely. Because the mother's immune system never sees the foreign antigen, it never becomes sensitized, it never builds its own permanent memory B cells, the synthetic antibodies we injected degrade naturally over a few months, leaving the mother safely unsensitized for any future pregnancies. We preemptively trick the immune system into ignoring the breach.
Science is just amazing. Speaking of amazing science, there is a section in the research that points toward a futuristic solution to all these transfusion incompatibilities. A way to bioengineer universal blood.
It's a fascinating area of study.
We established that type O is the universal donor because it naturally lacks the dangerous A and B sugars. What if we could just manually remove them from type A and B blood? Give the cells a microscopic haircut.
It is a stunningly brilliant concept that biochemical engineers have been actively pursuing for decades. If blood group antigens are ultimately just carbohydrate structures attached by highly specific chemical linkages, like the alpha-1,3 linkage we discussed, then theoretically we just need to find the right molecular scissors to snip those specific bonds.
And the material highlights the use of recombinant human glycoside hydrolases.
Right, glycoside hydrolases.
Scissors that cut sugars.
Exactly. Researchers began by looking at nature. Certain strains of bacteria in the human gut have actually evolved to digest human blood group sugars off the intestinal lining for energy. Scientists isolated the specific bacterial enzymes responsible for this, sequenced them, and bioengineered highly purified versions.
So theoretically, you could take a donated pint of type A blood, which normally you could only give to a type A or AB patient, and you introduce this specific hydrolase enzyme into the bag.
Yes. You incubate the blood with the enzyme. The enzyme acts purely on the alpha-103 and acetylgalactosamine bond. It races through the bag of blood, snipping the A sugars off every single red blood cell membrane, leaving only the bare H antigen foundation behind. The type A blood is artificially chemically converted into universal type O blood.
If they can refine and scale that process, the implications for global healthcare are almost incomprehensible. You would completely eradicate the concept of blood shortages in trauma centers. You wouldn't need to panic if you run out of O negative. You just enzymatically convert your surplus inventory of type A and B. What is holding this back from being standard practice right now?
The primary hurdle is absolute biochemical perfection. You need 100% cleavage efficiency. If the enzyme misses even a tiny fraction of a percent of the millions of A antigens on those cells, the remaining antigens could still trigger a fatal hemolytic reaction in a sensitized recipient.
Right, the immune system is too sensitive.
Exactly. Furthermore, you have to engineer a way to completely wash the bacterial enzymes out of the blood before transfusion. Otherwise, the patient's immune system might mount a massive allergic response to the foreign bacterial protein itself. But the enzymatic efficiency is improving drastically, and clinical trials are pushing this closer to reality.
Okay, I want to dramatically shift our perspective now. We have been living deep inside the microscopic world, examining single molecular bonds, genetic mutations, and cellular architecture. I want to zoom all the way out and look at the entire human species.
Okay.
Because we casually toss around the terms ABO and RH as if they are the only relevant blood categories in existence. But those are just the tip of a massive biological iceberg.
That is perhaps the most common misconception regarding blood. As of the most recent updates compiled by the International Society of Blood Transfusion, or ISBT, there are currently 48, some counts say 49 fully recognized human blood group systems.
48 different systems.
Yes, and within those complex genetic systems, there are over 600 distinct, molecularly characterized antigens decorating the human red blood cell.
600 unique structures. And the ISBT basically assigns them all a specific 6-digit barcode, right? Just to keep track of the chaos.
Yes, the nomenclature has to be incredibly precise to maintain global scientific consistency. The first 3 digits of the barcode represent the blood group system, and the last 3 identify the specific antigen in the chronological order it was discovered.
Okay, give me an example.
So the ABO system is officially designated 001. The A antigen was the very first discovered, so its official global barcode is 001.001. The B antigen is 001.002. The RH system is system 004. But beyond those, you have dozens of other completely independent systems. The Kell system, Kidd, Duffy, Diego, Colton, Lutheran, MNS. Each of these systems represents completely different genetic loci with their own unique protein structures, transport channels, or carbohydrate chains.
Before we dive into what all those obscure systems actually do physically, the research outlines a fascinating historical detour. A massive scientific rivalry in the 1940s regarding how we even name and understand the genetics of these systems. Specifically, the battle between the Fisher race theory and the Wiener theory regarding the RH system. What was the central conflict there?
It was a fundamental, highly contentious academic dispute over how the complexity of the RH system was inherited at the DNA level. In the 1940s, we didn't have DNA sequencing. We just had inheritance patterns from families and reactions in test tubes.
So they were guessing.
Educated guessing, yes. Ronald Fisher, a brilliant statistician, and R.R. Race, a serologist, proposed what became known as the CDE nomenclature.
Which sounds straightforward enough. C, D, and E.
It was very elegant. They theorized that the various RH antigens were controlled by 3 distinct, separate, but closely linked genes sitting next to each other on the chromosome: a C gene, a D gene, and an E gene. Each gene produced one specific antigen, so you could inherit a D, a small c, and a large E.
It makes intuitive sense. 3 genes, 3 distinct proteins, 3 antigens. But Alexander Wiener disagreed.
Strongly. Wiener, who had co-discovered the Rh factor with Landsteiner, violently opposed the Fisher race theory. He proposed the Rh nomenclature. He argued passionately that it was genetically impossible for 3 separate genes to be linked that tightly without occasionally recombining and scrambling.
Okay, so what was his alternative?
Instead, he theorized there was only one single massive gene at one specific locus. This single gene produced one massive aggregate protein complex, and on that one protein were multiple different factors or binding sites that acted as antigens. To describe this, he used an incredibly complex notation system involving superscripts, subscripts, and apostrophes like R1, R, rho.
It sounds like a typographical nightmare. So who ultimately proved to be right? When we finally got the technology to sequence the actual DNA decades later, who won the debate?
The irony of scientific progress is that molecular genetics proved them both partially right and profoundly wrong. The reality was a bizarre hybrid of their theories. There aren't 3 separate genes, as Fisher race insisted, but there isn't just one single gene, as Wiener dogmatically argued.
What's actually there, then?
There are exactly 2 highly homologous genes situated directly next to each other: the RHD gene, which produces the D antigen pillar, and the RECE gene, which produces a single protein pillar that somehow carries both the CC and E epitopes folded onto it. Today, global blood banks use the Fisher-Race CDE letters simply because they are vastly easier to read on a medical chart and communicate in an emergency. But the underlying genetic reality is a fascinating compromise of both historical viewpoints.
That is a great story of scientific ego colliding with biological reality. Okay, let's look at the global map of these blood types. The data sets in our research detailing evolutionary geography are astonishing. When you map out the prevalence of blood types across different continents and populations, it is absolutely not a random distribution.
Not at all. The global distribution of blood types is perhaps the most profound living record of human migration, population bottlenecks, and relentless environmental pressure. Globally, if you average it out, type O positive is the dominant blood type. But when you look at specific clusters, the numbers skew wildly.
Like,
For instance, in countries like Peru and Ecuador, the prevalence of type O blood is routinely over 70%. If you test certain isolated indigenous populations deep in the Amazon basin of South America, the prevalence of type O approaches 100%.
why such an extreme concentration?
It reflects two things: incredibly strong indigenous ancestry and a massive genetic bottleneck effect. When early human populations migrated across the Bering Land Bridge into the Americas thousands of years ago, it was a small founder population. If those founders disproportionately carried the O allele and the environment didn't punish them for it, that gene became locked in as they expanded across the continent.
But then you cross the Atlantic and look at Europe, and type A suddenly spikes dramatically. It heads up to 50% in places like Austria, Norway, and Denmark. And the research points out an incredible statistical anomaly in North America. The Blackfoot Indians of Montana have an astonishing 80% prevalence of type A blood. It's the highest recorded concentration in the world, existing in an isolated population right next to populations that are predominantly type O.
And conversely, type B is heavily clustered in Central Asia, particularly among Chinese and Indian populations, where it can reach 25 to 30%. Meanwhile, AB negative remains the rarest blood type across virtually all global demographics, often sitting at less than 1% of any given population.
So the million-dollar question here is why? Why this intense geographical disparity? Is it purely just random genetic drift from populations migrating and isolating themselves, or is there a functional reason? Did the environment actively favor the type O broken gene in South America and the type A gene in Europe?
While genetic drift plays a role in isolated groups, the broad global patterns are almost certainly driven by intense natural selection. Blood group antigens are not just passive ID tags, they are the literal interface between our cells and the outside environment. Pathogens, viruses, bacteria, parasites use these specific sugar and protein structures as chemical handles to grab onto and invade our cells.
Ah, so it's an arms race.
Exactly. And the prevailing scientific theory explaining the massive global dominance of the type O blood group centers entirely on humanity's deadliest historical foe: malaria.
Malaria. That makes complete sense, given the staggering death toll it has inflicted throughout human history. Walk me through the mechanism. How does a single missing sugar on a red blood cell protect you from malaria?
It's a fascinating structural defense. Severe malaria is caused by the Plasmodium falciparum parasite transmitted by mosquitoes. When this parasite enters the human bloodstream, it physically invades the red blood cells to reproduce. Once inside, the parasite does something insidious. It fundamentally alters the red blood cell.
How so?
It forces the cell to produce sticky parasitic proteins, primarily one called PFEMP1, and push them out onto the cell's outer membrane.
It's turning the red blood cell into a sticky weapon.
Precisely. These sticky proteins cause the infected red blood cells to physically clump together, a process called rosetting, and adhere aggressively to the endothelial walls of the blood vessels. This rosetting causes severe, fatal blockages in the microvasculature of the brain, lungs, and other organs. This is what kills the patient in severe malaria.
And how does blood type intervene in this rosetting process?
Extensive clinical studies have demonstrated that the Plasmodium falciparum parasite binds much more effectively and forms much tighter rosettes when the A and B sugar antigens are present on the cell surface. The A and D sugars essentially act as superior biological grips for the parasite's sticky proteins.
So A and B are worse for you?
Yes. Red blood cells coated with A or B sugars form massive, tight, highly destructive clots. But type O red blood cells, which lack those specific A and B sugars, offer a very poor grip. They form much smaller, looser, more fragile rosettes that are easily broken up by normal blood flow.
So the parasite can still infect a type O person, but it can't create the fatal brain blockages as easily.
It's exactly. As a result, individuals with type O blood have a significantly higher statistical survival rate when infected with severe malaria. In malaria-endemic regions like sub-Saharan Africa and tropical parts of South America, the evolutionary pressure was immense. Over thousands of years, individuals with A and B blood died before reproducing at much higher rates. The environment heavily, relentlessly selected for the type O gene. The molecular pathogens of the ancient world were the actual sculptors of our modern blood type demographics.
That is profound, and it perfectly transitions us into the final major exploration of this dive. Blood type as a deeply functional molecule and a predictor of chronic disease. Because we've established that these antigens are structurally vital, like the Rh pillars preventing the cell from bursting, and we've seen them as evolutionary shields, like type O surviving malaria.
Right.
But what are all these 600 plus antigens actually doing right now mechanically in a healthy listener's body? They aren't just sitting there waiting for a mismatched transfusion or a malaria-infected mosquito.
Far from it. The research heavily categorizes these 600 antigens by their active biological functions. It turns out your red blood cell membrane is an incredibly busy piece of biological machinery. A vast number of these obscure blood group antigens are actually vital transmembrane transport channels, constantly moving molecules in and out of the cell.
Give me a specific example of how this works.
Let's revisit the Rh system. We talked extensively about its structural role anchoring the membrane, but the specific RhCG complex within that system has a highly active functional role. It acts as an intricate channel, specifically designed for transporting ammonium and carbon dioxide across the lipid bilayer, which is vital for helping the blood regulate its pH levels.
Okay, what else?
Another system is the Colton blood group. The Colton antigen is not a random protein, it's actually aquaporin-1.
Aquaporin, a water channel.
Yes, it is a massive, highly specialized pore responsible for rapidly transporting sheer volumes of water in and out of the red blood cell in milliseconds, allowing the cell to instantly adjust its internal volume and maintain osmotic balance as it travels through different salinity gradients in the kidneys.
That's incredibly specific.
And the Diego blood group antigen is physically located on band 3, which is the cell's primary anion exchanger. It constantly swaps chloride ions for bicarbonate ions, facilitating the massive transport of CO2 from your deep tissues back to your lungs to be exhaled.
I really want you, the listener, to grasp this. These obscure blood types, Colton, Diego, Rh, they aren't just arbitrary medical ID tags. They are the actual plumbing, the ventilation shafts, and the water pumps keeping your cells alive second by second.
That is the perfect way to contextualize it. And beyond transport channels, another major functional category for these antigens is adhesion molecules, or CAMs. Red blood cells don't just float passively, they need to chemically interact with their environment.
Like Velcro.
Exactly. For example, the Indian blood group antigen is located on a massive receptor complex called CD44. CD44 acts literally like molecular Velcro. It actively binds to hyaluronin, collagen, and fibronectin in the extracellular matrix. It helps anchor immature red blood cells in the bone marrow during erythropoiesis, the actual creation process of blood.
But there is a dark side to this cellular stickiness, right? The material highlights how these adhesion molecules play a devastating role in sickle cell anemia.
Yes, unfortunately. In sickle cell anemia, the core issue is a mutation in the hemoglobin inside the cell, causing the red blood cells to undergo a pathological physical change, becoming rigid and sickle-shaped. But a secondary, highly destructive consequence is that the cell's biochemistry also alters. The surface antigens shift their behavior. The cells become hyperadherent. They stick aggressively to a specific structural protein called laminin that lines the walls of the blood vessels.
So the sickled cells aren't just getting physically stuck in the pipes because of their shape. They are chemically gluing themselves to the walls.
Exactly. And this abnormal adhesion induces pathological signaling changes in the vessel walls themselves. It causes the endothelial cells to retract and upregulate procoagulant molecules. This hyperadhesiveness, driven directly by the altered behavior of the surface antigens, is the primary driver of the devastating vaso-occlusive crises in sickle cell patients, where the blood vessels completely clog, causing ischemic tissue damage and immense pain.
So the antigens are actively, aggressively participating in the disease process, which directly connects to the massive American Heart Association journal study highlighted in our research. The AHA published incredibly robust data showing that your basic ABO blood type is statistically, undeniably associated with your lifetime risk for cardiovascular disease.
It is a stunning, heavily verified statistical correlation. The AHA study analyzed millions of patient data points across multiple decades. They found that individuals possessing Group A and Group B blood are statistically associated with significantly higher baseline risks of hyperlipidemia, deep vein thrombosis, which is dangerous, spontaneous blood clotting, and myocardial infarction, meaning heart attacks.
We have to unpack the why here. I don't want to just give you all a scary statistic without context. Why do A and B sugars on a red blood cell make your heart more likely to fail? What are those sugars mechanically doing?
The prevailing physiological mechanism links the ABO blood group directly to the plasma levels of von Willebrand factor. Von Willebrand factor is a massive, complex glycoprotein that is absolutely crucial for the blood clotting cascade. It essentially acts as the primary glue that binds platelets together to seal a wound.
Okay, so it's the body's natural wound sealant.
Right. Now, clinical assays show that individuals with non-O blood types, so types A, B, and AB naturally, have significantly, sometimes 25 to 30% higher baseline levels of von Willebrand factor and factor VIII circulating in their plasma compared to type O individuals.
Why do they have more of it?
Because the von Willebrand factor itself is actually decorated with ABO sugar antigens. The A and B sugars seem to chemically protect the von Willebrand factor from being quickly degraded and cleared by the liver. Because it sticks around longer in the bloodstream, the overall levels remain much higher. As a direct result, the blood of type A and B individuals is inherently, functionally more prone to clotting.
Which, in an ancient hunter-gatherer environment where you are constantly at risk of being mauled by an animal or suffering severe traumatic bleeding, having blood that clots incredibly fast is a massive evolutionary superpower.
Precisely. It was a survival advantage.
Hmm.
But in a modern, highly sedentary population with cholesterol-rich diets, that ancient superpower translates directly into a higher risk of spontaneous thrombosis and heart attacks.
But the type of folks listening right now shouldn't be celebrating entirely, right? They aren't entirely off the hook.
No, biology always extracts a trade-off. While the study confirmed type O individuals have a notably lower lifetime risk of spontaneous clotting and heart attacks, the data found they are statistically associated with a higher risk of developing severe hypertension or high blood pressure. The exact molecular mechanism for that specific correlation is still being intensely researched, but it perfectly demonstrates that every genetic adaptation in the human genome comes with a physiological cost.
Okay, I have been eagerly waiting to dive into this next specific detail because it is, without a doubt, the most mind-bending science fiction concept in the entire stack of material. We have to talk about the parasite Toxoplasma gondii.
Yes, the notorious feline parasite. It is a masterpiece of evolutionary manipulation.
Right. Listeners, I need you to stay with us here. Toxoplasma gondii is a microscopic, single-celled parasite that typically reproduces exclusively in the intestines of cats. Humans generally catch it by accidentally ingesting the oocysts from handling infected cat litter, or by eating undercooked meat from livestock that grazed on contaminated soil. Once inside a human, the parasite migrates to the brain and forms dormant cysts in the neural tissue. For decades, the medical consensus was that this latent infection was completely asymptomatic in healthy humans. But recent, incredibly robust neurological studies show it actually alters human behavior.
It is a textbook example of neuromodulation. The parasite has evolved the ability to subtly, continuously alter neurotransmitter levels in the host's brain. Specifically, the parasite's genome encodes an enzyme called tyrosine hydroxylase, which is the exact enzyme needed to manufacture L-dopa, the precursor to dopamine. The parasite is literally pumping dopamine into specific regions of the host brain to alter behavior.
In mice, the outcome is famous. The dopamine flood makes the mice lose their innate evolutionary fear of the smell of cat urine. In fact, it makes them attracted to it. They essentially offer themselves up as easy prey, which allows the parasite to get back into the cat's intestine to complete its life cycle. It's hijacking their brain. But humans aren't prey for house cats, so what does it do to us?
In human hosts, massive epidemiological studies have linked latent toxoplasma infection, which affects roughly one-third of the global population, to distinctly altered behavior. Infected humans show a statistically significant increase in generalized risk-taking behavior, measurable delays in neurological reaction times, and remarkably, a statistically robust higher incidence of being involved in severe traffic accidents.
Which is an utterly insane concept on its own. A microscopic brain parasite from a cat subtly rewiring your dopamine pathways to make you crash your car. But how on earth does a red blood cell type factor into this neurological hijacking?
The researchers diving into this behavioral phenomenon found a stunning, highly specific statistical correlation with the Rh blood group system. They ran extensive reaction time tests and behavioral surveys on massive cohorts of infected military drivers and civilians. They discovered that Rh-negative individuals who were infected with Toxoplasma gondii exhibited profoundly impaired reaction times and a massively increased risk of traffic accidents compared to uninfected individuals. The parasite's neuromodulation was highly effective on them.
But what about the Rh-positive infected people, the ones with the massive D-antigen pillar bolted to their cells?
The data showed they were largely protected. The Rh-positive individuals carrying the exact same parasitic brain cysts did not demonstrate the same severe cognitive impairment, dopamine dysregulation, or delayed reaction times. The effects were vastly mitigated.
So you were telling me that the RHD protein, that massive structural pillar we talked about holding the red blood cell together, is somehow acting as a neurological shield against the mind-controlling effects of a brain parasite? How is that mechanically possible?
That is the profound implication of the data, and it opens up an entirely new frontier of research. The prevailing hypothesis suggests that the RHD molecule might play an entirely unrecognized vital role in the central nervous system. It isn't just structural. Perhaps it plays a critical role in modulating localized oxygen transport or rapid ion exchange in the neural tissue under the specific physiological stress induced by the parasite. Or perhaps the Rh complex fundamentally alters the baseline immune response to the cyst in the brain, changing how the localized inflammation affects the dopamine receptors. We don't have the exact chemical pathway mapped yet, but it is a completely unexpected functional link between a blood group antigen and complex neurobiology. It forcefully reminds us that these molecules are integrated into our biology in ways we are only just beginning to map.
It makes you wonder what else these antigens are quietly doing in our brains. Okay, one final massive tangent, and this brilliantly brings everything full circle to where we started. We talked at length about the secretor system earlier, the FUT2 gene that instructs your glands to spit your specific ABO sugar antennas out into your bodily fluids and the mucosal lining of your gut. The final papers in our research stack merge this concept with metabolomics and the human microbiome.
This is truly the bleeding edge of current physiological research. Metabolomics is the exhaustive, comprehensive study of all the small molecules, metabolites, and chemical processes within a biological system. And currently, researchers are aiming the immense analytical power of metabolomics directly at the human microbiome, the trillions of symbiotic bacteria currently living in your intestinal tract.
Right, and we generally accept that our diet, whether we eat high-fiber, high-fat fermented foods, radically alters which strains of bacteria thrive in our gut. But the research explicitly states that our blood type is an equally powerful, genetically hardwired architect of our microbiome. Explain the mechanism there.
It is considered one of the most significant, immutable genetic host factors shaping the microbial landscape. Think back to the mechanics of the secretor system. If you are part of the 80% of the population that is a secretor, your gut lining is not just a passive barrier. It is constantly, actively secreting a thick mucus layer that is absolutely saturated with your highly specific A, B, or H blood group sugars.
Oh, wow. I want the listener to really internalize this concept. The trillions of bacteria currently thriving in your stomach and intestines right now are actively, selectively feeding off the specific sugar antennas determined by the chromosome 9 blueprint you inherited from your parents.
Exactly. You are literally farming different bacterial colonies based entirely on your blood type. The bacteria in your gut are in a constant, brutal competition for resources. Some bacterial strains, like certain species of bifidobacterium, have evolved highly specialized enzymes to perfectly cleave and rapidly consume type A sugars. If you are a type A secretor, you provide a massive, constant food supply for them, and their populations explode.
But if I'm a type B secretor,
Then you are secreting B sugars. Those specific type A-loving bacteria will fundamentally starve. Their populations will dwindle and die out in your gut, while entirely different strains of bacteroids that specifically possess the enzymes to digest type B sugars will flourish and dominate the ecosystem. Your genetically determined blood type physically dictates the baseline nutrient environment of your intestinal tract.
the implications of that are staggering. It means there is no such thing as a universal healthy microbiome.
Precisely. It has massive immediate clinical implications for personalized nutrition, the development of targeted probiotics, and understanding the root causes of inflammatory bowel diseases like Crohn's. It even dictates how we metabolize oral medications. Different bacterial profiles break down drugs differently. The blood group antigens are the literal physical interface between our human genetic code and the vast microbial world living inside us.
What an unbelievable journey. Okay, let's take a breath and summarize exactly what we've unpacked today. We started with the common assumption that blood types were just arbitrary letters on a medical chart, but we've zoomed all the way in to see them as dense, intricate forests of specific sugar trees meticulously constructed by highly specialized enzymatic robotic arms. We've seen the RH system as massive multi-pass protein pillars bolted directly to the cellular skeleton to prevent the cell from exploding under pressure. We've mapped out the rigorous genetic math of Mendelian inheritance and explored how the secretor system paints these specific molecular structures throughout our mucus, saliva, and bodily fluids.
We examined the catastrophic violent immune responses dictated by Landsteiner's law, tracking the complement cascade as it punches holes in foreign cells. We analyzed the terrifying genetic missing pieces that create the Bombay and Raynaud phenotypes. We covered the historical tragedy of maternal-fetal incompatibility and the prophylactic miracle that solved it. And we looked at the incredible futuristic bioengineering efforts using bacterial enzymes as molecular scissors to snip sugars and artificially create universal blood.
And finally, we mapped these antigens across human history and the globe, seeing them not as static markers, but as dynamic evolutionary armor against pathogens like severe malaria. We saw them acting as functional plumbing and water channels for the cell, as statistical predictors of clotting and heart disease, as completely unexpected neuroprotectants against mind-altering feline parasites, and as the very foundational food source shaping the microscopic ecosystems within our own guts. It is an absolute, sprawling microscopic universe inside every single drop.
It is a profound testament to the immense mind-boggling complexity hiding just beneath the surface of the mundane everyday biology we take for granted.
Which leaves me with a final provocative thought to mull over as we wrap up. If our blood type is basically a living historical record of our ancestors' brutal evolutionary arms race against ancient diseases, with pathogens like malaria constantly evolving new sticky proteins to dock onto our specific cell surface receptors, and our ancestors only surviving because of genetic deletion mutations like type O, what invisible modern environmental pressures are currently silently acting upon us. With the rise of entirely new zoonotic viruses, exposure to novel synthetic chemicals, and radically shifting global diets altering our microbiomes, are we still evolving entirely new blood systems right now, slowly rewriting the genetic barcodes beneath our own notice?
From a biological standpoint, it's a certainty. Evolution never actually stops. It simply operates on a timeline and scale that we cannot easily observe within a single human lifetime. The pressures are always there, selecting the survivors of tomorrow.
Definitely something to think about the next time you get a paper cut or fill out a form at the doctor's office. We encourage you to view your next blood draw not as a routine medical chore, but as a glimpse into your own microscopic, deeply personal evolutionary universe. Thank you for joining us on this deep dive.